I will tell you about our study on notch functions in the corneal epithelium. Our general interest is trying to understand how self-renewing tissues are maintained. What kind of signaling pathways maintain stem cells as stem cells and which one drives them into differentiation. And we use mostly mouse genetics to study this. And we're interested in evolutionary conserved pathways because it has been shown that although they have important roles during embryogenesis, they are also very important for the homeostasis of self-renewing tissues. And so from our studies working on notching in the skin, we know that loss of notch 1 in the skin has very important roles. However, we did not know whether this is also important for the corneal epithelium. Now, the difference between the skin and the corneal epithelium is that the cornea is a non-stratified, non-keratinized stratified epithelium. That is very important to assure actually transparency of the cornea. It is composed of a basal cell layer that has pluristratified squamous cells on top of it. It is separated from the stroma by a basement membrane, and the stroma is actually the largest part of the cornea. It contains keratocytes that produce lots of collagen that are organized in parallel bundles that assure actually transparency. And so the epidermis or the cornea is particular because it is avascular. So it means it has no blood vessels, and that is important to ensure good vision and so that light can really go through.
So by using the transgenic mouse models that were available in the lab at the time, it was possible to delete notch 1 in the corneal epithelium, and this could be done either during development or postnatally. And when this was done, what was found was that the mice developed opacities on the ocular surface. And when this was analysed histologically, what was found was that the corneal epithelium resembled a hyperproliferative epidermis, i.e. something that you would normally find in skin. And in fact, the phenotype was very similar to what you find in mice which have been deleted for notch 1 in the skin. So this suggested that in the absence of notch signalling, what happens is the corneal epithelium undergoes a fate switch to the epidermal lineage. So to confirm this, immunohistochemistry was performed using markers specific for epidermis, such as keratin 1 and loricrin. And when this was done, it was found that indeed these markers were expressed by the cornea, thus confirming that a fate switch had occurred from cornea to epidermis. So this kind of fate switching phenotype is consistent with the classical notion of how notch signalling operates. So when the mice were analysed in more detail, it became clear that this probably wasn't how notch 1 was functioning in the cornea. So firstly, however notch signalling controls corneal identity, it does so specifically in the context of wound healing, so it isn't required for the normal homeostasis of the corneal epithelium. And secondly, and most importantly, the fate switch which occurs in the epithelium is a non-cell autonomous event. So what happens is, prior to any kind of cell fate switch, the underlying stroma undergoes extensive remodelling and modification, and it's only once this event happens that you get the conversion to epidermis in the overlying epithelium. So the results from this study led to the model that was proposed in the publication, which was basically describing how notch functions in the cornea. So during a normal wound healing response, you have the epithelium, which is repaired by corneal epithelial stem cells resident in that tissue. A notch in these cells really functions to control the crosstalk that occurs between the epithelium and the underlying stroma. However, when you ablate notch signalling, as we did with the mouse models, the regulation of this crosstalk is perturbed, and the notch deficient epithelium induces changes in the underlying stroma, which leads to this extensive remodelling, and it's this remodelled neostroma which instructs the epithelium to differentiate into epidermis.
All right.
The tubulin.
Tubulin is normal, exactly.
Normal. Exactly.
That is a cool result. If
Yeah.
phase 1 is gone, that's... The long-term goal, obviously, of this study is to use this knowledge in order not to make skin out of cornea, but to do the reverse, to use skin stem cells in order to be able to make cornea, because that would have a big therapeutic impact.